One of the main mechanisms for obtaining energy is cellular respiration, in which glucose molecules are degraded to produce ATP. The process begins in the glycolysis, then the Krebs cycle, and then the electron transport chain. This evidence will mainly highlight the production of CO2, water, and ATP. Glycolysis takes place in the cytoplasm, especially in this process for ATPs are produced. In this process, it is known that the glucose begins to break down by different enzymes involved. The importance of the point is when fructose 1,6-biphosphate is divided in two, when 1,3-biphosphoglyrate is reached. The use of ADP is necessary here. The first two of the base of the glycolysis are produced. It continues until reaching phosphoenol pyruvate. When the same process occurs, another two ATPs are produced, and then the cycle continues until the production of two pyruvates is reached. Two NHD are produced also. Subsequently, pyruvates enters in the Krebs circle. The Krebs circle takes place in the matrix of the mitochondria, and in the matrix of the mitochondria, this process is also explained once, but it's understood that two of this happens is two pyruvates enter to the cycle. Pyruvate is converted to acetyl-CoA by pyruvate dehydrogenase, producing a CO2 molecule. Later in the cycle, in the isocitrate, the enzyme isocitrate dehydrogenase is used, obtaining alpha-ketoglutarate and producing another CO2 molecule, followed by the enzyme alpha-ketoglutarate dehydrogenase, producing succinyl-CoA, which produces another CO2 molecule. By means of succinyl-CoA synthase, the first ATP of the cycle is produced, obtaining succinate. Later in the cycle, it continues and it's repeated again. It should be known that in this process, 8 NADH and 2 ADH are produced, but for the purpose of the video, the steps obtained were omitted. In the electron transportation chain is the last part of this process. We obtain ATP by using NADH and FADH2. The first complex receives NADH to transport two hydrogen ions from the matrix to the space in the intermembrane space of the mitochondria. Passes to complex 2, which through the coenzyme Q pumps two protons out of the matrix. In the fourth complex, two protons are pumped in the intermembrane space so that an oxygen atom obtains two electrons and other protons to obtain a molecule of water. For FADH2 molecules, this process is the same, and only they leave the electron directly in the coenzyme Q. Subsequently, the proton concentration gradient between the intermembrane space and the matrix of the mitochondria produces potential energy, which is used for the synthesis of ATP. For passing through the ATP synthetase, two protons activate and the enzyme, and this is catalyzes the synthesis of ATP from ADP molecule and phosphate group. This process produces 3 ATPs for every NADH and 2 ATPs for every FADH2. And for every 2 electrons in the complex, where molecules are obtained. Lately, in the results of this process, we received 36 ATPs, and this is the process of the cellular respiration. Thank you.